Tēnā koutou, and welcome to another biochemistry highlight for the day. Remember, the material is 100% non-examinable. Intrigued by the PDP resources for coronavirus last week, over the long weekend, I looked at their PDB-101, which includes mini diversions based on macromolecular structures. Here's the home page. It offers both informative and decorative looks at protein structures. Above the fold, as it were, is their current feature. In this case, it's the photosynthetic reaction center, which sits under the phycobilizome in red algae or among the chlorophyll antennae in green plants. And so that's always worth checking out. Over on the right down here, our particular interest to us today are virtual backgrounds for Zoom meetings. So you can choose from myoglobin, ferritin, or the nucleosome for your background for your next virtual Zoom meeting. Perhaps not quite as decorative as palm trees on a tropical island, but quietly geeky for your next Zoom meeting. Back to their main homepage and down at the bottom are galleries from two of the great illustrators of molecular structures, Irving Geis and David Goodsell. You'll certainly recognize Geis's illustrations from your earliest introduction to hemoglobin, for instance. He had a genius for letting the functional shine through a structure and making it eye-catching at the same time. And so you almost certainly remember that from Biochemistry 192. Finally, at the very bottom of their homepage here are the little links to animations and videos illustrating particular things, and my personal weakness, paper models. If you're lucky enough to be in lockdown with a laser printer, you can print these out and make them yourself in a escape from perhaps writing a Bioc 351 lab report. Making the viral capsids is a good way to think about the laws of symmetry, which govern both how viral capsids assemble and how crystals grow. This weekend, I made both insulin, thinking back to Dorothy Hodgkin and the Festival of Britain, and the double-stranded DNA one. The product of my playing around is shown up here behind me. Insulin doesn't look like very much on paper, in my opinion, but I was very pleased with how the double-stranded DNA came out. You can clearly see the phosphodiester backbone, the major groove, and the minor groove, showing that this is much more open on this side. I think Odile Crick would have approved of the representation. Poke around in the PDB and see what else there is in there besides the archival structural files.